In this session, we study more about logistic regression, with focus on its regression coefficients, or beta coefficients, as adjusted odds ratio. By studying materials of this session, we will understand the meaning of e to the beta, as the adjusted odds ratio, in the logistic regression model, and we will be able to interpret estimated values of e to the beta in logistic regression results. This is the outline of the lecture. First, we study how to interpret logistic regression results in terms of odds ratio. Then we will go through some simple examples that help us to understand the mechanics of logistic regression, and then look at some published examples. Let's study the odds ratio in the context of logistic regression. This is the result of a logistic regression analysis from the SAS example in the previous session. The table is titled Analysis of Maximum Likelihood Estimates. The dependent variable was self-reported general health and independent variables are gender, age, race/ethnicity, education, and marital status. Since the logistic regression model includes a linear combination of independent variables, the beta coefficient for each independent variable was estimated, just as in linear regression. For example, the estimated beta coefficient for age is 0.0206, and that for divorced or separated is 0.4955. These two estimates will be used as examples in later slides. But beta coefficients in logistic regression are difficult to interpret. 0.4955 means that log odds of being self-reportedly unhealthy for the divorced or separated is estimated to be greater than that for the currently married, which is the reference category, by 0.4955, controlling for gender, age, race/ethnicity and education. But log odds is an unfamiliar quantity to us. What does a 0.4955 difference in log odds mean? It is not intuitively clear at all. The SAS output has another table, titled Odds Ratio Estimates, which displays a quantity that is easier to interpret than beta. The column labeled point estimate shows the exponential function of beta, that is, e to the beta. For example, the estimate for age is 1.021, which is e to 0.0206, and the estimate for divorced or separated is 1.641, which is e to 0.4955. And if we request confidence intervals, SAS shows 95% confidence intervals for e to the beta, instead of confidence intervals for beta. Why is e to the beta useful? Because it can be interpreted as the adjusted odds ratio. It is adjusted in that the analysis controls for the other independent variables. It is about odds of the event of interest. In this example, it is odds of self-reporting fair or poor health. And it is the ratio between the category of interest and the reference category. Another major table in the SAS output of the logistic regression is labeled Type 3 Analysis of Results. The rightmost column shows p-values for numerical and categorical variables. The same p-values for numerical variables such as age, and dichotomous variables such as gender, are shown in the table titled Analysis of Maximum Likelihood Estimates shown 2 slides before. But overall p-values for polytomous variables such as race, ethnicity, education, and marital status, are shown in this table of Type 3 Analysis of Results, but not in the table titled Analysis of Maximum Likelihood Estimates, which was shown 2 slides before. The earlier table shows p-values for binary variables, corresponding to categories of the polytomous variable, but not p-value for the overall categorical variable. Now, let's go through the basics of the odds ratio. To those who have taken an introductory epidemiology course, this will be just a review. But it may help to have an overview of regular odds ratio, i.e., unadjusted odds ratio, before talking about adjusted odds ratio. Relative risk and odds ratio are general statistical tools that can be used in any field, but they are widely used particularly in health research for measuring effects of risk factors when we are interested in the risk of a particular disease. In general, relative risk and odds ratio are statistical indicators for measuring the association between two dichotomous variables. In health research, usually one of the two variables is a risk factor that has two categories, exposure to the risk factor, and no exposure to the risk factor. The other variable may be disease incidence, with two categories indicating whether the person developed the disease or not. The other variable may also be disease prevalence with two categories that indicate if the person has the disease or not. Relative risk, also called risk ratio, proportion ratio, or probability ratio, is the ratio of two proportions. Because a proportion is also an estimate of a probability, it may be interpreted as the ratio of two probabilities. In epidemiology, It is the ratio of two proportions between two groups. The proportion of interest is the proportion of those who develop the disease or the proportion of those who have the disease. Two groups are those who were exposed to the risk factor and those who were not. Odds ratio is similar to relative risk, but it is the ratio of two odds and not two proportions. Odds is the ratio of two probabilities, the probability for the event to occur and the probability for the event not to occur. Let's look at an example, focusing on relationship between age at first birth and breast cancer. Now, it is widely known that late childbirth and no childbirth raise the risk of breast cancer. Data on childbirth and breast cancer were collected from 6,168 women in middle age and old age. The investigators classified early first birth and late first birth as births to women under age 25 versus births to women aged 25 and over. 1,628 women had their first childbirths at age 25 and over, and 31 among them developed breast cancer, but 1,597 did not develop breast cancer. For the late first birth group, the proportion of those who developed breast cancer is 31 over 1,628, and the odds of developing breast cancer is 31 to 1,598. Now, 4,540 women had their first births before their 25th birthdays. 65 among them developed breast cancer, and 4,475 did not. For the early first birth group, the proportion of those who developed breast cancer is 65 over 4,540, and the odds of developing breast cancer is 65 to 4,475. Let's copy these numbers to the top of the next slide. The relative risk of breast cancer between the two groups is 31 over 1,628, to 65 over 4,540, which is 1.330. This means that the proportion of breast cancer incidence for the late first birth group is 33% higher than that for the early first birth groups. The odds ratio of breast cancer, between the two groups, is 31 to 1,597, over 65 to 4,475, which is 1.336. This means, the odds of breast cancer incidence, for the late first birth group, is 33.6% higher than that for the early first birth groups. Note that the relative risk and odds ratio are quite close, and the odds ratio is slightly higher. This is usually the case for low probability events such as the development of breast cancer. The majority of women do not develop breast cancer during their lifetime, though the incidence rate is relatively high in comparison with many other cancers. So, the relative risk and odds ratio are based on similar statistical ideas. And the relative risk may be easier to interpret than the odds ratio, because we are usually more familiar with the concepts of proportion and probability than the concept of odds. But the odds ratio is widely used in health research because it is important in case control studies. In a case control study, data are collected from people who developed the disease and people who did not. Those in the two groups are selected in such a way that the two groups are comparable with respect to characteristics other than the disease. Usually those who develop the disease are oversampled because statistically these types of diseases are rare events. The proportion of those who develop the disease is small. Therefore, the incidence proportion and the odds of the disease in the sample are usually much higher than the actual proportion and odds, which also makes the relative risk unreliable. But oversampling does not bias the odds ratio. Let's look at this hypothetical example. This table is similar to the one we just looked at. In this data, those who develop breast cancer are oversampled, 10 times more cases than in the previous table, 310 women, instead of 31 women, who had their first births at age 25 or over developed breast cancer, and 650 instead of 65 women who had their first births under age 25 developed breast cancer. So, of course, the proportions and odds of breast cancer computed from this table are much higher, and the relative risk is 1.282. Which is lower than 1.330 in the previous table. But the odds ratio, which can be computed as 310 times 4,475 divided by 650 times 1,597, is 1.336, the same as that in the previous table. Therefore, the odds ratio is quite convenient in health research, in which we often need to oversample disease cases in case control studies.